Okay, so today we're going to be discussing biotechnology, which is basically the way we are able to study genes within organisms and use organisms to produce things with genes. Not only genes, but also proteins and RNA and other properties of molecular properties of cells. So the reason why we're able to do this is because of techniques that involve recombinant DNA. Recombinant DNA is DNA from two different sources found within the same organism. Um, and the reason why we are able to do that is because of enzymes we've discovered called restriction enzymes. Restriction endonucleases is what they're also called. And what they do is they find specific sequences of DNA and they are able to cut those sequences. We originally discovered these because bacteria use them to cut apart the DNA of viruses, and in that way making the viruses unable to infect them. There are three types of restriction enzymes. Type 1 and 3, they cleave not at in less predictable patterns than type 2, and so we use type 2 to as a tool in making recombinant DNA. When they are cut at specific sequences, they don't cut evenly down the middle. They cut and separate these sequences so that you have these sticky ends. Sticky meaning there is only one base pair on each side, so all you need is another base pair to come in and bind there. Okay, and then those ends, so if you have another section of DNA with that corresponding sequence, you can insert those and they will then bind to them. And DNA ligase then is the enzyme that actually goes in there and seals the nicks or binds them all together. Okay, and you can see that in this animation here. You have the sticky ends with corresponding sequences that go together. They bind together, but in order to fully be formed, ligase has to come in and bind the phosphodiester bonds of the backbone of the DNA. Okay, so the bonds between the phosphate and the hydroxyl groups between the DNA nucleotides, which we all know forms the phosphodiester bonds. This is the same enzyme that binds together the Okazaki fragments on lagging strands in replication. So we have experience with gel electrophoresis, which separates DNA, or in lab we separated proteins based on their charge and size. And they go through a gel made of agarose or polyacrylamide, submerged in a buffer that can relay the current. We turn the power source on, which creates the current, making a positive and a negative end, and then the negatively charged DNA or proteins or RNA or whatever we're looking at will migrate towards the positive pole. The larger fragments move slower, and the smaller fragments are going to move faster, and so you can separate and find differences based on the size of the fragments. The DNA is made visual because they put on fluorescent dyes or other. When we use the protein, we had dyes within the proteins to make them more visible. Okay, so another technique is called transformation, where you introduce foreign DNA into a cell. And this is a natural process in many species, but we use E. coli because it naturally does not do this. So to do that, we induce the process of transformation by either warming them up or cooling them down. And that allows us to insert DNA into the E. coli. There's also another molecular method called molecular cloning, which is making genetic identical copies of certain genes and being able to insert them or not, or take them out of organisms. Usually the DNA that we are looking for to clone is one that codes for a protein, and this is also sometimes called gene cloning. Again, we use E. coli. A little bit about E. coli. It is naturally found in the gut of most animals, including humans. It's a bacteria, very simple bacteria, and it lends itself easy to replication and DNA insertions. So in order to get DNA in there, you first have to have a vector, which is something that will carry the DNA into the host. And the host vector system, depending on the function of the gene, can have a particular use. So one of the vectors that we use very commonly is called a plasmid, which is basically like a miniature chromosome. It's a small circular chromosome which is in excess to the single intertwined chromosome that bacteria usually have. There are three components to the plasmid. You have the origin of replication. Okay, so if you want to make new copies of the plasmid, that's where it would happen. You have a selectable marker, which allows the plasmid to be identified, and then you have cloning sites, or multiple cloning sites. Alright, artificial chromosomes, sometimes the plasmids are too small for the gene of interest, and so sometimes we use yeast or bacteria as the vector. And if we use yeast, it's called yeast artificial chromosome, or YAC. And if we use bacteria, it's bacterial artificial chromosome, or BAC. And these are just larger, better able to insert larger genes or a host of genes within an organism. A method that has been used quite a bit extensively since the late 80s, early 90s is called polymerase chain reaction, developed by Kary Mullis and received the Nobel Prize for it. What it does is it allows you to use very small amounts of DNA and amplify it, so then you can figure out what the whole host of DNA in there. Okay, what you have to do is you first have to denature the DNA or get it to pull apart. You can see that in this picture here. You give it to expose it to a high temperature, and it will pull apart. And then it will be annealing of primer, so that means putting a primer on there, usually an RNA primer, that will bind to these strands that you pulled apart. And the third one is adding enzymes like DNA polymerase that will synthesize the other strand. Okay, if you do that once, you'll end up with 2 copies or 4 copies of DNA. You do that again, you end up with 8 copies, and so on and so forth. So you do this over and over and over again, you have lots of copies of the DNA where you originally only had 1. The DNA enzyme that we use is called Taq polymerase, and the discovery of this was very important to the invention of PCR. So the application of this is you really don't need very much DNA. You can just have it replicated in multiple copies, and then you'll be able to better analyze the DNA. So this is valuable with forensics or with defects in embryos, so you don't need a lot of DNA there. There isn't a lot of DNA there. Also with historical archaeological finds of human species or other animal species, you can get DNA from a very little bit. Another important method is called making a DNA library, and this is a collection of DNAs within plasmids and then they're inserted into bacteria, and this represents a genomic library within all the bacteria that you have. What happens is the genome is randomly fragmented. This is going to be done using restriction endonucleases. And they insert it into a vector. In this case, is a plasmid, and then introduced into host cells. And then they keep these cells or these cell lines in storage and then you have your plasmid library. You have your set of DNA within these organisms. Another very similar one is called complementary DNA or cDNA. It's where you make DNA copies of mRNA. Okay, mRNA, when it makes proteins, though, it doesn't have introns. The exons have already been taken out. And then an enzyme called reverse transcriptase, what it does is it takes mRNA and then reverses the copy back into DNA. And then that is called cDNA, and then that is used to make a library. Okay, it's not the same as the original DNA, again, because the exons have been taken out. And then to visualize DNA, there are a few methods. Southern blotting is where DNA is digested by restriction enzymes, cut up into little pieces, and then run through a gel electrophoresis. And in order to do this, the double-stranded DNA has to be denatured into single strands, and then it will be blotted by filter paper, so the filter paper will pick up the DNA, and then also given a probe. A probe will bind to those single strands and will cause it to fluoresce or give it color so that you can then see the DNA. Northern blotting is basically the same process, only you're looking at messenger RNA, and Western blotting is the same thing, but looking at proteins. So you may see in movies or videos or even documentaries where they're doing methods of science, methods with DNA. You'll see these sheets, and they're usually plastic sheets, full of these lines and bars of amplified DNA. This is either southern, northern, or western blotting techniques. Okay, DNA fingerprinting. This is just as you might think. Fingerprints are used to as unique signatures for humans. This is done using what's called restriction fragment length polymorphisms, or which are different or unique to all humans. Basically, there are certain points in the DNA that are slightly different, and so they will have point mutations, or sometimes a duplication of a sequence will be found. Okay, and if you go through a person's DNA, you will find lots of differences. They'll have duplications, which will make for a longer fragment, or you will have base pairs, where the restriction endonucleases will cut, but because this base pair is different on this other person, it's not cut there. So it will make all these different sizes of DNA. The different sizes will correspond to the uniqueness of the individual. Okay, and then again, we use southern blotting to visualize those. So medically, this is all important because we can use then bacteria and the enzymes within them to make human products, including insulin, vaccines, and the biggest deterrent to this has been being able to purify the desired proteins or make enough of them in a small amount. So these are an example from your book where genes are used to make A and B chains of insulin, and then once you get those together, it makes our active insulin that can be used for diabetics. Vaccines are also used. We use bacteria to make them. Subunit vaccines have genes that encode for a part of the protein coat specific to a disease, for example, cowpox. You inject this into a human, and we are able to make the antibodies necessary for immunity. DNA vaccines are also made, but they depend on the cellular immune response. So we're not looking at immunity there. Okay, we still have about 10 slides left, and I'm about to run out of time, so we'll stop here and then pick up on the next YouTube video.